and welcome to our series, Science and Society, Making Sense of the World Around Us. This lecture series is co-organized and moderated by Fred Dilla, Executive Director Emeritus of the American Institute of Physics and author of Scientific Journeys, Linda Dilla, former Public Information Officer at the Jefferson Laboratory in the U.S. Department of Energy, and Colin Norman, the former news editor at Science. And with that, I would like to turn this over to Linda. So Linda, please remember to unmute yourself. Make sure you can unmute it there.
Here, I'm unmuted. Hi, everybody. I'm so glad you all are interested in this topic. Dr. Don Lawrence, or as we call him Don, we've now known him almost 9 years because he's the oncologist who cured my husband of stage 4 melanoma. The word cure is an interesting word to use as far as melanoma is concerned. There was a time when it was a death sentence and you just gave someone 6 to 9 months to live, unless you wanted to start cutting things off and you could make it last longer than that. But there wasn't a cure. In Fred's case, I think we can almost brag that he has one. We never say that, but Don is the reason that that's the case. And this man is a lecturer at the Harvard Medical School, and he's the clinical director for the Center for Melanoma at Mass General in their cancer center. And we go see him every 6 months. And I can't tell you how much I've learned from him. I still read abstracts from his profession, and what he's doing is at the top of his field. So I'd like Don to tell you about what he's doing now that is even more cutting edge than what was done to get Fred to the point where he is now.
Thank you, Linda, Fred, Colin, the Lewis Library, everyone for joining in. And I'm not really going to talk about anything that I personally have done. I'm going to try to give a very quick run through of about 5,000 years of medical history, oncology history, and then we'll focus on the last 50 years. And what I see is the major trend, the major area of progress, which is the move towards personalization of treatment and how that's come about and where it's going. So great to be here. Let me share my slides. There we go. Slideshow. Okay. Can you see my slides? Is that everyone with me? So personalized cancer treatment, personalized medicine gets a lot of lip service, and I'm never sure what it means. I think we talk about it a lot more than we see it. We still basically treat all of our patients the same. The standard of care has really not changed much, but I do believe it is changing, and that change is based on an understanding of the fundamental biology of cancer and other diseases, and the employment of some incredible scientific breakthroughs, technology that are now making their way into the clinic. So with that, so here's the previous, 4 9/10 millennium of cancer treatment, surgery, evidence archaeologically of cancer surgery going back to 3000 BCE. And then really oncology got its start in the early 20th century with radiotherapy. I tend to ask questions rhetorically as I go, and so, but please don't come off mute, but you can put your answers in the chat if you want, if anyone knows who this person is, this woman in the middle, who was really the inventor of radiotherapy for cancer. You can chime in the chat, but I'll just tell you, it's rhetorical. That's Marie Curie. who ultimately died of bone marrow aplasia from exposure to radiation. But her observations in the lab very quickly led to the therapeutic use of radiation for cancer treatment. And then chemotherapy, I was surprised to learn this in my training. It is a relatively recent invention. The idea that you could treat cancer with drugs is really, only within the last 80 years or so has been possible. And in fact, we can date to the the month, year, month, day, and time when the first chemotherapy was given. And so these three modalities have been referred to as cutting, burning, and poisoning cancer. But I think that really does a disservice. This represents a just enormous research effort to bring these types of treatments to patients, to refine them, and to use them in combination to improve cure rates for cancer. And the paradigm of this is in breast cancer, where surgery used to be radical mastectomy, very deforming surgery. The emphasis has moved to less and less surgery, using radiation as an adjunct to surgery, and chemotherapy, hormonal therapy, and now other newer biologic therapies to reduce the risk of relapse. So, just a remarkable achievement over many decades. International research has led to the cure of many women who otherwise would have died from breast cancer and other cancers. But we're not so hot at treating cancer when it's metastasized. That's a whole different ballgame. And chemotherapy can be curative, but in a tiny number of cancers. Testicular cancer is number one. High cure rate with chemotherapy. We could talk about why, but we won't. Lymphomas, leukemias. Beyond that, the common solid tumors, lung cancer, colon cancer, all of them. The drugs have had a palliative effect, but do not generally have curative potential, and that is still the case today. So I think that the story that I want to tell really starts with these two goofy-looking British gentlemen on the left and their colleagues on the right, Watson, Crick, Wilkins, and Franklin, who discovered the structure of the genetic material, DNA, which immediately suggested its function as the transmitter of genetic material from one generation to the next, and also its function encoding proteins, which determine all of our biologic traits, all physiologic functions. So these guys were awarded, I say guys, because we'll see next, three of those people were awarded the Nobel Prize in physiology or medicine. One was not, and that was Rosalind Franklin. So that's a whole other story that I won't get into. So the next leap forward in terms of cancer is the recognition, or the sort of the inkling that cancer is a disease of disordered genes. And the early research in the 70s actually identified genes that were inserted into cells by viruses that would cause a disorder in the growth process of the cell, leading to transformation, meaning turning a normal cell into a cancer cell. And initially this led to the idea that many cancers, or most, were caused by viruses. That's not the case. It's actually fairly uncommon. But what is and remains true is that these damaged or different genes, which we call oncogenes, they create mutated proteins, and specifically proteins that drive the growth of cells and push them towards malignant transformation. Uh, so some of these gene abnormalities are inherited, such as the BRCA gene, which predisposes to breast cancer. Rarely they can be introduced by viruses, but most of the oncogenes probably arise as a result of DNA damage from carcinogens, tobacco, in the case of lung and other cancers, ultraviolet radiation in the case of skin cancers. So cancer is a disease of damaged DNA. I hope there are no Nobel Prize winning molecular biologists in the audience, because I'm in a world of trouble if they are, but onward. Uh, so again, to skip ahead another 20 to 30 years, from the identification of the structure of DNA and the insights into its function as the genetic material, uh, we arrive at just an enormous undertaking, the Human Genome Project, to map all 20,000 genes in the human genome. And basically this is to tell us what the normal array of genes is in human beings. And what took 15 years at the time, we can now do in days with the current technology, map individual genomes, whole exome sequencing. These processes have become automated and can be done rapidly at scale and with reasonable cost. So we do have the capacity now to really take a deep dive, not only into the sort of generic human genome, but the individual human genome. And as a subset of that process, uh, what has emerged is the Cancer Genome Atlas, which is looking at the genomic changes, the mutations in normal DNA that occur in different cancers. So again, now very high throughput, rapid analysis, and large scale, 33 different types of cancers, over 10,000 samples, identifying the array of genetic abnormalities that distinguish tumor from its host. So these mutations can be divided up into a couple of different categories, and much of the focus for the last 20 years has been on what are called the driver mutations. These are the relatively few mutations that play a direct role in stimulating or actually, causing a cancer or leading to accelerated growth, progression, resistance to programmed cell death, which contributes to cancer. So these are the driver mutations because they drive the growth, and they become very important therapeutically, as I will get to, uh, and really have led to a different nomenclature to describe cancer. We still refer to lung cancer, colon cancer, etc., site of origin and appearance under the microscope, adenocarcinoma, squamous cell carcinoma, large and small cell, different descriptors that are now kind of quaint, actually. But the evolving definition of cancer recognizes that what looks the same under the microscope can vary drastically at a molecular level. And so the key questions now are not only, is it lung cancer, and what type of lung cancer, but what are the gene mutations, what are the driver mutations that are key to its growth, its spread, its survival. And based on this this thought process, I'm using the example of lung cancer here. Numerous driver mutations have been identified that are key in the pathogenesis of lung cancers and that differ from one individual to the next. And in lung cancer, these are relatively uncommon. There are mutations found in a minority of lung cancer patients, and sometimes a very small minority, and they include things, genes such as EGFR, ALK, et cetera, all listed here. So we can divide more and more finely the cancers into their molecular signatures. So this is interesting. Why is it important? Well, it's important because of the development of targeted therapies. These are small molecules that get inside the cancer center, the cancer cells, and are capable of shutting down these growth switches that are stuck in the on position. So these are inhibitors of these specific altered proteins that are made by these altered driver genes, these mutated genes. And these have been amazingly successful but for only a relatively small percentage of lung cancers. For example, the EGFR mutation was the first driver mutation identified and targetable with medicines, but it accounts for only 15% of lung cancer, although much higher in Asians. And it tends to occur in younger people, females, and non-smokers. So the 10% or so fewer of lung cancers that do occur in non-smokers, they tend to have these driver mutations that are targetable with drugs. And these drugs are amazingly effective. They work and they shut down the growth switches, they kill cancer cells, and in some cases can lead to very prolonged control of disease. Are they ever curative? That is, I don't know the answer to that question, not being a lung cancer specialist, but my sense is although they can lead to control for an extended period of time in advanced lung cancer, that they ultimately lose their effectiveness, their resistance emerges, and the cancer progresses. Uh, Now, in my area, in melanoma, we don't have the same array of different gene abnormalities that we've identified that drive the growth. We really have a few. And by far the most important is a gene called BRAF, which it's in a signaling pathway. Again, it's a circuit that is stuck in the on position, and we have drugs that can break that circuit. These are oral therapies. They're reasonably well tolerated. They're not chemotherapy. And the theme continues, as with lung cancer, in that these are very effective in the short term, but almost inevitably resistance emerges. That's a whole other subject. What are the mechanisms of resistance? And so for advanced melanoma, metastatic melanoma, these are excellent palliative treatments and can be life prolonging, but are rarely, if ever, curative. And then for the 50% of melanomas that don't have BRAF mutations, uh, other driver mutations exist, but we're not so good at developing drugs to target them. RAS has been a really intractable target in many cancers, different RAS isoforms. That the code there seems finally to be cracking with the development of drugs that can target RAS mutated proteins. So just an example, this is from the very first publication on the BRAF inhibitor vemurafenib, the first to be developed. This was published by my colleague Keith Flaherty a number of years ago now. And these are PET images of patients with widely metastatic melanoma. taken before the start of BRAF inhibitor therapy. Those are the images on the left, and their corresponding images on the right are after just 15 days of treatment. And I don't think you need to be a radiologist to sort out what's going on here. The patient, for example, in the upper left, this is his or her liver completely infiltrated with cancer. That's the blackness, and then all that's gone in 2 weeks. Similarly here, a sort of smattering, a Jackson Pollock pattern of tumors, and all gone metabolically cold after 2 weeks on the PET, and so on. So when we first saw this in the early trials, you can only imagine the excitement, and but also the disappointment that these responses were transient, and that as well and as quickly as people responded and felt better, they would inevitably relapse with, the average time is a little under a year, although that's quite variable. So targeted therapy has a major role in the treatment of these cancers and is much more effective than chemotherapy, particularly in melanoma and also in lung cancer where those driver mutations are found. But it's not the answer. But it is a sort of lead in to the idea of truly personalized medicine in dividing cancer into finer and finer subcategories based on an understanding of the molecular mechanisms that make them tick, and really the recognition that if you look deep enough and hard enough, everyone's cancer is different from anyone else's cancer. And if we can crack that code and really dive more deeply into the entire array of dysregulated genes, then we may be able to make progress towards cure. So I would look at this as miraculous, but preliminary progress that has happened over the last 20 years or so that sets the stage for further refinement of individualized cancer treatment. Okay, so, well, I think I said most of this already, but targeted therapy advantages is that it is personalized based on the mutational profile. High response rate can prolong survival. These are generally oral treatments and can be taken as maintenance therapy. When an oncologist tells you that the side effects are manageable, just reach for your wallet, because manageable is in the eye of the beholder. These can sometimes be difficult, but I think definitely better than chemo in that regard. So downsides, as I said, only a minority of lung cancers, for example, have targetable mutations that are amenable to these kinds of treatments. Half of melanoma does, a BRAF mutation, but for those who don't have that BRAF mutation, there's really no good targeted therapy, and not curative in advanced disease. So that I would say is the state of the art now, but it's a field that's in rapid flux and development. So a word about resistance, which is a theme of targeted therapy. Why does resistance emerge? And this also goes to the notion that not only is one person, my cancer is different from my brother's cancer or anyone else's at a fundamental level, but also that even within an individual patient, there is heterogeneity of the cancer cells, and what we call clonal diversity or clonal evolution. The cancer may start out from one cell that was transformed, that went haywire, but it then develops along a kind of evolutionary pathway with different clones and subclones that acquire different capabilities and ultimately some that are able to evade the effects of targeted therapy. And the same goes for immunotherapy. We see the resistance of the emergence of resistant clones in immunotherapy as well, although at least in melanoma, not as frequently. It's almost universal with targeted therapy in melanoma. With immunotherapy, we do feel that we can eradicate the disease completely despite the clonal evolution in some patients, maybe up to half. So up till now, I've focused on these driver mutations, which are relatively uncommon in terms of the whole landscape of altered genes in cancer, and only are found in a small minority of lung cancers. About half of melanomas have the BRAF mutation. And if you go down the list of other cancers, it's a pretty small percentage that have driver mutations that are targetable by drugs. But in the background of all of this, and ignored for quite a while, were all these other mutations that occurred. Again, these are uh, gene alterations that arise largely from carcinogens, from ultraviolet radiation, from tobacco, other things that mess up your genes. And so they don't only cause these key mutations in genes that regulate growth. There is a much larger number of these passenger mutations. They have really nothing to do with driving the growth of the cancer. They're just there, and there are many of them. And until recently, they were kind of the poor stepchildren of mutations, thought of as random junk. But a clue as to their importance came from this kind of analysis, which actually really predates the modern era of immunotherapy, in that melanoma is on the far end of the what we call the mutational burden, the number of mutated proteins in a melanoma cell is higher than in virtually any other cancer. And some of the other cancers on the far right edge of this figure also are ones that are the most responsive to immunotherapy. So it took a while to put this story together, but it turns out that these random passenger mutations are very important for immunotherapy. No impact on the function, growth, death of the cell, but they make a cancer cell look different enough from normal self that the immune system has a chance of recognizing it as foreign. And that is what the immune system has evolved to do, recognize self versus other and attack other, mostly infectious organisms. So while cancer is self, it's derived from us, these passenger mutations can make it look different enough that it can elicit an immune response. And the greater the number of those genetic mutational events, the more likely it is that something is going to be recognized by the immune system. And so, well, with that said, it turns out that the immune system can recognize cancer in many, many cases as not right, not belonging there, not normal self, but it really can't do anything about it. And this gets to one of the other central themes of immunotherapy, which is that the cancer actually actively suppresses the immune response. It co-opts normal mechanisms of immune quiescence to evade the immune system. If it did not do that, most cancers would be spontaneously, many would be eradicated by the immune system. So, as a sort of Darwinian process, they have evolved mechanisms to block the immune system from attacking them. And the current generation of immunotherapy drugs disables these mechanisms of immune evasion. These are drugs that many people, they're familiar with the names because they advertise them on TV, things like pembrolizumab or Keytruda, nivolumab or Opdivo. These are PD-1 antibodies. PD-1 is one of these key brakes on the immune system. And by interrupting, by taking off that brake, releasing it, that is sufficient in many people to lead to a profound anti-tumor immune response and eradication of metastatic cancer in some people with melanoma. So this is what I've told you is, of course, a gross oversimplification. There is a lot that the immune system has to do to recognize a cancer cell as non-self, to mount an immune response, to the T cells of the immune system have to traffic through the body and engage with the tumor cells. So many points at which the immune response can fail. And to me, it's somewhat remarkable that one little intervention, the blockade of this PD-1 brake, is sufficient to lead to such a profound immune response. Uh, but it works. It doesn't work in everyone, and why not? And how to help the people in whom it doesn't work, that is at the cutting edge. Okay, this is just graphically what I was describing in terms of PD-1 blockade, taking a break off the immune system and allowing the T cells of the immune system to attack. So just as a graphic example of this, this is a patient I met now over 3 years ago. He came into the emergency room. Sorry, the scans are flipped. The one on the right is the earlier scan from November 9th, 2021. And again, you don't have to be a radiologist to see that on this brain MRI, there are three enormous tumors. Uh, and with this gray haze around them, that is edema, that is swelling. So this patient came in with stroke-like symptoms and had these massive tumors in his brain. Prior to the development of the current generation of immunotherapy drugs, we would have been talking about hospice right then and there in the emergency room. But because we knew we had a chance with this patient, we took a very aggressive approach, not me, but one of my surgical colleagues, resected uh, these three areas. And so his postoperative scan shows things look better. These are the postoperative changes, but he still had other tumors in his brain and in other locations and elsewhere in his body too. So this is an example of what, on a different level in the brain, he had two not quite so huge, but pretty large tumors as well. So he was treated then with a combination of these what are called immune checkpoint inhibitors, these drugs that take the brakes off the immune system. And within 4 months or so, this is his corresponding brain scan at the same geographic level. You can see a little flush of the contrast in the area where that tumor was, in the frontal lobe, and the one in the occipital lobe is basically gone. I saw him last week. He's doing fine. He stopped racing his race cars, which was his hobby, but otherwise he's doing well. I just, in some ways, I just have to show off, but really it gives you a sense of the power of these treatments, even under very desperate circumstances where the prognosis would have been a matter of a few weeks before the existence of immunotherapy. So to get to the theme, is immunotherapy personalized? It is and it isn't. So it's certainly, it's more nuanced than chemotherapy. It's more effective than chemotherapy for many cancers, but it's still kind of what I would call off the rack. We are, in some cases, distinguishing patients in advance who may or may not benefit from immunotherapy. In melanoma, regardless of molecular profile or other testing, it is our best treatment. And so we do not withhold it, although we know certain patients are less likely to benefit. But in lung cancer and other cancers, there are clearly patients who are just not going to respond, and you don't bother treating them. But we have really only a few choices. There are a few immune checkpoints, a few breaks on the immune system that we are capable of releasing. PD-1, CTLA-4 is another. LAG-3 just entered the arena, and then one that was promising called TIGIT. The clinical trial just was reported as negative. So we don't have that many tools, and we're trying to apply these tools across the board and then to understand retrospectively why they work well in some people and not at all in others. But it's not a prospective process at this point, which it really needs to be. I haven't talked about the toxicity of these treatments. They're not like chemo, but they can have very substantial, serious side effects. So it would really be nice to know who will benefit and who wouldn't, but we're not there yet. So other forms of immunotherapy that are in current practice, oncolytic viruses, we actually can elicit an immune response by injecting an attenuated herpes virus into melanoma tumors, which sounds terrifying, but it can work. And then there are molecular constructs, including CAR T cells, which is much too complicated and it's too large an area to go into. But these are like designer immune cells, but their use has really been limited to the hematologic malignancies, lymphomas, leukemias, multiple myeloma up until now. And they've not really made their way into the treatment of solid tumors. So I think the personalization will come in a more nuanced use of these drugs, developing other drugs that target other checkpoints, and really recognizing the complexity of resistance to immunotherapy and how to manipulate the tumor microenvironment to advantage to turn some of these non-responders into success stories. Okay. So the one of the latest things in melanoma is actually a different kind of immunotherapy, which is really rather ancient. This tumor infiltrating lymphocyte therapy was developed at the National Cancer Institute by Steven Rosenberg literally 40 years ago. And we've known for a long time that this can work in some patients. It really takes a different approach, using the patient's own immune cells, their T cells, as in essence a living drug to treat the cancer. So this involves removing a tumor, not for curative purposes, but to obtain the T cells, the immune cells that are inside it. They are there because presumably they are recognizing something that doesn't smell right about the tumor and trying to mount a response, but they're not doing a good job. Otherwise, the tumor wouldn't be there. However, if you isolate these T cells and grow them up to large numbers, culture them, you can create literally billions of these cancer-fighting, melanoma-fighting cells, which are called tumor-infiltrating lymphocytes. It's not only the sheer number, though, that makes them go, it's also the fact that they are taken out of the immunosuppressive tumor microenvironment, which I mentioned before, that the tumor actively kills T cells, puts them to sleep, deactivates them, and when you remove them from that environment, they wake up, become more active, more able to mount a response. So you have more and better melanoma fighting T cells. They are then reinfused into the patient, just like getting a blood transfusion, and they make their way back into tumors and attack them under the best of circumstances. That's not all there is to it. It's actually a very toxic treatment because people need to get chemotherapy prior to the cell infusion. Again, not to treat the melanoma, but to sort of pave the way for these immune cells. If you don't give the chemotherapy, the cells just disappear. So you have to kind of ablate the rest of the immune system to make room, so to speak, for the TIL cells. And then patients get a drug called interleukin-2 after the infusion. This interleukin-2 is a T cell stimulator growth factor. It's a naturally occurring, what's called a cytokine, and we give it in doses that are far in excess of what the body would normally produce. And it mimics sepsis, severe infection. So it's not a pleasant drug either. So this is a bear of a treatment and requires hospitalization and is only for the fit and the brave. But when nothing else works, when all our other immunotherapy treatments fail, we can see durable remissions with TIL therapy. We've known this for many, many years from the National Cancer Institute, that there is what we would call a tail on the curve. These are, just bluntly, the people surviving over time, and quickly a lot of people die, but then over time there are about 20% who are long-term survivors after TIL therapy. So the recent innovation, this was brought to market by a company called Iovance, was really not a scientific innovation. It was the ability to create TIL to go, I would call them. We take out a tumor from a patient, we ship it directly from the OR to their lab. They isolate the T cells, they grow them up, and then 5 weeks later, they send us back a bag of TIL. To do that on your own, even at an institution like Mass General, is an enormous logistical and regulatory undertaking. There are only a few sites around the country that are able to generate their own TIL, although that is on our agenda to do that. So shifting gears now, again, let me go back and tell you why I included this in a talk on personalized cancer medicine. This is getting towards personalization because we're using the patient's own cells. So obviously those are unique to that person. They would not work in another person because they would be rejected by the immune system, but also because these cells are reacting to the unique mutations in that person's own melanoma, which may be and probably are very different than the mutations in somebody else's melanoma. So even if you could give this to the next person, it probably wouldn't work. My T cells would not attack your melanoma because there are different mutational profiles. So it is getting at that personalization. What is kind of strange about this, though, is that we don't know what the targets are, the molecular targets in the melanoma that are eliciting the immune response that allow the TIL to work. We just gather the TIL, We don't know what they are recognizing or attacking. We put them in and let nature sort things out. And then if we're lucky, we have a clone or population of T cells that recognizes antigen, a protein, an abnormal protein on the melanoma cells, and kills them. So this is in a way personalized medicine, but it's also a black box in some other ways. So a further step towards personalization is, and this is something that's made an enormous splash, I think deservedly so, because it's a technical feat, just unbelievable that this could even be done. It's like science fiction to me, but it's kind of outstripped what we know clinically about the benefits of personalized cancer vaccines. So the idea is that we can identify those abnormal proteins on or in the cancer cell that are created by these mutated genes, identify the ones that are capable of eliciting an immune response, and then essentially use those proteins or the RNA that codes for them as a vaccine. And the platform is very similar. It's basically identical to the COVID vaccine, where you get an RNA, a messenger that codes for the spike protein in COVID. And therefore your cells produce the spike protein, which is a foreign protein. The immune system recognizes it and becomes primed to attack anything else that has a spike protein, and that namely is the COVID virus. That's the way the vaccine works. This is more complicated because it's not simply one protein and it's not a foreign protein. These are mutated self proteins, and the current platform allows for the identification of around 30 different proteins or peptides that are mutated and that are immunogenic. And this goes through so it requires sequencing the entire DNA of the tumor, sequencing the normal DNA, seeing what's different, and then a kind of computational analysis to predict what is immunogenic and also what will fit with the patient's own immune system. So this is, just unthinkable to me that this can be done in real time to prepare a vaccine for a real live patients within a few weeks, and that it actually seems to be promising. So very low toxicity. The excitement in melanoma was based on a relatively small but randomized study. And this was not in people with advanced melanoma. This was in people with high risk stage 3 melanoma, where we generally use pembrolizumab or Keytruda to lower the risk of those patients developing advanced disease, metastatic disease. And pembrolizumab works pretty well. We know that. But the addition of the vaccine to the pembrolizumab for these patients led to a much higher percentage remaining free of their melanoma over a 2.5-year period. That's where things stand right now. It's a proof in principle that this can work. And right now, that's enough for me. I mean, stage 3 melanoma is not that common. Studies are also being done in lung cancer and other cancers. This is going to be transformative. And this is the first indication that it can work. But I think it's just the beginning. And when it starts getting applied to for patients with more advanced cancer, it's just going to be tremendous. So I'm a believer in this, but it's not quite ready for prime time yet, although I think it will enter clinical use before too long in high-risk melanoma. So I think I've come almost to the end of what I wanted to say, except to point out that, just the theme is individualized, personalized medicine. But the theme also, I have to admit, is gross oversimplification, even based on what I've told you about gene mutations that differ from one person to the next, and everyone's tumor is different, but also every host is different. Every person, their immune system, their microbiome, meaning the bacteria that populate their intestines, has a profound effect on the immune system. So, there's a convergence of different ways of studying not only the tumor, but the person who has the tumor that is really going to allow a full flourishing of personalized cancer medicine. And this is being done, but it's a huge undertaking. And I'd say we really are at the outset of this kind of integration of information about different systems. All right, I will stop there and I'm happy to take questions.
We have questions. Thank you, Dawn. That is so interesting. And I think that gut biome piece is mentioned in literature frequently. It seems like the healthier you are when you get something like this, there's some connection that we just don't understand. They're talking now about the gut being the mind of the body or something.
I'm not sure what the metaphor is. All I can tell you is that there is a surprising and powerful connection between the bacteria in our intestines and our immune systems, and probably our central nervous system and many other systems as well. But just briefly, I'll tell you that if you take a mouse and give it melanoma and then treat it with this type of immunotherapy, and it responds and the tumor dies and the mouse lives, and then you have another mouse that didn't respond under the same circumstances, and you take the poop from that first mouse that was successfully treated and give it to the second mouse, then that mouse will then respond to the immunotherapy. It's that powerful. And, giving a human someone else's poop is, it's a little complicated, but there is a thing called fecal transplants where people can take tablets of stool that has certain properties that promote an immune response. I think ultimately we're going to be doing this with a much more refined way of identifying the bacteria that are helpful and creating mixtures of that. But the science is just unbelievable that it is demonstrating this connection. I wish I knew how to take advantage of that. I don't think probiotics are likely to be the path. If there is something out there that might be beneficial, it's a lot of fiber, which promotes a favorable gut microbiome.
I've got some of the best questions here. I'm going to pull a couple of them together, but this is one that I think is so true. At least some of these newer treatments seem to involve early or earlier detection in order to be the most effective. What can be done or is being done in that area?
Yeah, so that's a fantastic question. A lot of what I've described is playing catch up. It's once a cancer is metastasized, it's a miracle that we can treat it successfully at all, and we can in some cases. But far better is to catch it early. So the area of early cancer detection is also being influenced by this individualized, patient-centered approach. I mean, that's a whole other talk for someone else, not me, to give for 3 or 4 hours. But A very active area of research. I mean, right now, we do screening colonoscopies and mammograms to try to catch early cancers. But really, by the time something is evident on a colonoscopy or a mammogram, you've already got cancer. Can we detect signs of cancer in blood tests, in molecular analysis of stool, these are really where the field is going, and that is going to lead to fewer patients developing advanced cancer. So absolutely, far better than all this miraculous immunology to treat advanced cancer is to catch it early and prevent it from growing.
Oh my goodness. Well, here comes, this is from one of our friends who's a doctor. So hopefully, Jim, you did this in a way I can help everybody else understand it. In an
New England Journal of Medicine.
Yeah, sorry. In 2021, MNRA was used as a vehicle to deliver HSV1 to childhood brain tumors that were high-grade gliomas. Is this kind of immunotherapy being used in adult cancers? Because you did mention HSV1 and melanoma, he believes.
Yeah, so it is. I think its use is really limited. But these are what are known as oncolytic viruses, meaning cancer-killing viruses, and they get inside the cell, they proliferate, and they can be directly toxic to the cancer cell, but they can also evoke an immune response. And I did allude to one drug that is currently in use called TVEC, which is this modified herpes virus that can be injected directly into tumors. But it only works if you have a tumor that can be directly injected. And it can work not only on the tumor you inject, but on others nearby. But its impact in widely metastatic cancer is limited to nil. So the viral therapy of cancer it's an idea that's been around for a long time. I recall the publication you're referring to. I think there are going to be better ways of treating high-grade brain tumors rather than putting viruses into them. And in fact, CAR T cells are being developed to target brain tumors and injected directly into the cerebrospinal fluid. So I don't know. It's not my area of expertise, but it's kind of striking that that made such a splash, and then we haven't heard anything about it since.
Okay, one more question because we're coming up on to 6 o'clock. Number one, I want to tell everyone that we have Dawn's email, and if you have specific questions that are just killing you,
Or
please, or not killing you, but you just need them. Sorry, poor choice of words there. Email us. You have our email, so you can get to us, and we'll get them to you, to him. So the last question is, what other cancers do checkpoint inhibitors seem to work on? And are we finding more?
Yes, many, many, and more all the time. I've lost track of the number of cancers for which checkpoint inhibitors are now FDA approved, but they do not work as well in most of those cancers as they do in melanoma. Melanoma was like the first and foremost, and it was dramatically successful. It's been more incremental progress. Lung cancer, they can work, but not as well. And then there are many that are completely refractory, like pancreatic cancer, for example. So why that is, it has something to do with that concept of mutational burden. Just more mutations, the higher the chance that something is going to look funky to the immune system. And melanoma has a boatload of mutations, largely caused by UV radiation, but it's not that simple. There are other factors. So I can't give you a list, but there are many, many of them. At least 20 cancers, I think.
Thank you. Thank you for that.
Mhm.
Well, I hope you all in the audience agree with me that this is a topic that we can all take a little bit of information away with, take it to our friends and family, and use it for yourself if you need to. I will say to everybody who has any kind of condition, be your own patient advocate, look for the best that you can find, ask questions, ask for another doctor, do what you have to do to cure yourself or to make yourself better until you're satisfied. That's what works for all of us. All of us.
I'm a good example because I had a very good patient advocate and a very good oncologist looking after me. And on that note, I want to mention that Don mentioned the first radiologist was Marie Curie and the whole Curie family. And our talk 2 months from now, on April 13th, is by Dava Sobel, who just wrote a wonderful book on the whole Curie family, on her amazing lifetime that included many things other than what Don mentioned. And then next month, not to skip March, we have the world's expert on horseshoe crabs, a fine little fella that lives here in the Delaware Bay, and many of you probably know that they're very important for pharmacology. So stay tuned to the library, and we're going to turn it back over to Rebecca, who will let you say thank you to our speaker tonight.
Yeah, so as is our custom, if you would like to unmute yourself and applaud and say thank you, feel free to do so. So thank you.
Thank you.
Thank you.
Thank you.
Thank you so much, everyone. Thank you, Rebecca and Colin and Linda and Fred and everyone. And it's just tremendous to be able to participate in an event like this.